Hello, dear professors. Today we are going to talk about the inflammasome forms. It's me, Amal Mustafa Slim, Assistant Lecturer of Oral Biology, Faculty of Dentistry, Cairo University. The inflammasome is really important today to know, and let's first know something about the before we talk about the inflammasome, let's first know something about the innate immune system. The innate immune system engages an array of germline encoded pattern recognition receptors, or the PRRs. The PRRs are expressed by the cells at the front line of defense against infection, including macrophages, monocytes, dendritic cells, neutrophils, and epithelial cells, as well as the adaptive immune system. These PRRs can detect or act as the surveillance machine. And this one can actually, or this job can lead finally by activation of the inflammasomes and inflammatory response, finally to the death of this microorganism, or even this process can happen with the self-antigens. So this is a very important steps to be happening. These PRRs could be or can be actually DNA sensing or RNA sensing. So these PRRs that are membrane bound receptors can actually be very important for the inflammasome. So what are the PRRs? PRRs could be either one of three important forms that are the TLRs, or toll-like receptors, membrane-bound toll-like receptors, or CTLs, or C-type lectins, and these two types actually can scan the extracellular or endosomal compartments for the pathogen-associated molecular patterns. We got also the NLRs, the topic for us today, or the node-like receptors, which are a set of intracellular PRRs that are distinct actually from those two TLRs and TDLs, and these can actually recognize the PAMs, or the pathogen-associated molecular patterns, as well as the host-derived danger signals, or the danger-associated molecular patterns, or the DAMPs. These two actually are cooperating together to provide cytosolic surveillance, including the RNA sensing or DNA sensors. Actually, these PRRs, depending upon the nature of both the responding cell and the invading microbe, can lead to the outcome of the recognition by the PRRs. Here we focus on the PRRs that assemble into high molecular weight caspase 1 activating platform that are the inflammasomes. And especially actually we are going to talk in special about the NLR family, or the NLRs, or the node-like receptors. So actually, we first need to know more about, before we talk about their functions and components, we need to know what are the NLR family. What is it, and what are the subfamilies if it has. Well, the NLR family is comprised of 22 human genes, and it's actually having many more mouse genes because of gene expansion since the last common ancestor. This phylogenetic analysis of the NLR family, or having what's called the NACHT domain, or NACHT domain, reveals three important subfamilies, as we are going to see here in the next diagram, where we can see here the nodes, or NODs subfamily, that is containing node 1, 2, node 3, or NLRC3, the node 4, to the end of this subfamily, and the IPAF subfamily that consists of IPAF or NLRC4, and also we can see the third one, the NAIP subfamily. Also, these three subfamilies comprising a very important stuff here. Now let's see another diagram where we also can see the three subfamilies, the node subfamily and the IPAF subfamily, also the AIM2 subfamily, which is another one because it's not or not a node subfamily, but yet has an important effect here. So let's now move to what are the inflammasome popular forms. We got four important forms: the NLRP3 inflammasome and the NLRP1 inflammasome, and these two are very characterized today. The third one is the IPAF or NLRP4 inflammasome. Also, we got the AIM2 inflammasome. We are also at the end of this presentation. We're going to talk about other members of the NLR's family that are actually having also other functions to provide for us today. Let's move to the NLRP3 inflammasome. This one is currently the most fully characterized inflammasome, as I just mentioned earlier, and consists of the NLRP3 scaffold and the ASC or the PY card adapter and the caspase 1. These three important items for this one are really important. The caspase 1 can be recognized as the effector protein, and the ASC is the adapter protein, while the NLRP3 PYD card or the NACHT domain, where it's present at the core of it, is the platform protein. Actually, the NLRP3 is activated upon exposure to either PAMs or DAMs, but also it can be recognized and activated by the whole pathogens. Whole pathogens like fungi, bacteria, viruses, they all can activate the NLRP3. Fungi like Candida albicans, bacteria that produce the pore-forming toxins, including Listeria or Staphylococcus, and viruses like adenoviruses or even influenza virus. All these whole pathogens can activate finally the NLRP3. Now let's move to the next one, which is the NLRP1 inflammasome. This one is having actually three orthologs in mouth, while for that in the human, it also completely different than the NLRP3 counterpart, as we can see here, as the NLRP1 contains a C-terminal extension that harbors a card domain, as we can see here. And this one can interact directly with the pro-caspase 1 to the lower right part of the screen, and this one can actually interact directly with the pro-caspase 1 and bypass the recruitment of ASC, although ASC inclusion in the complex augmented human, the NLRP1 inflammasome activity. Actually, the IPAF inflammasome or the NLRC4 inflammasome is also very important, but it's activated by the gram-negative bacteria processing the type 3 or 4 secretion systems. The IPAF contains a card domain, and it interacts directly with the pro-caspase 1, which is also a very advantage for this one here. The maximal caspase 1 activation in response to certain types of bacteria actually requires the ASC adapter. This ASC adapter rule in the IPAF inflammasome actually still remaining unclear till now, but it's presumed that these proteins do not interact directly as IPAF doesn't have a PYD domain, and thus it's possible that the IPAF collaborates with a PYD containing protein such as the NLRP. So this is to or for response to these pathogens can have an ASC responsible for the IPAF dependent caspase 1 activation in response to these types of bacteria. But protection against these types of pathogens requires IPAF collaboration with another NLR family, that is the NAIP. So IPAF dependent caspase 1 activation is accompanied by rapid cell death after this one. Now let's move to the last one for today, which is the AIM2 inflammasome. Actually, this one is recently identified, and it's not a node family. It's a HIN 200 family member. It's a cytosolic double-stranded DNA or dsDNA sensor that induces caspase 1 dependent interleukin 1 beta maturation, and this is really important in advance of the inflammasome field. Actually, it's also the first to be identified as non-NLR family, forming an inflammasome scaffold, and oligomerization here happens in a different way than what we know for the NLR family through the NACH domain from the central part. But it happens by clustering around upon multiple binding sites in the ligand. This one is binding to which the AIM2 binds via its C-terminal HIN domain. It's also quite permissive that this one can be recruited as systolic DNA, double-stranded DNA from viruses, bacteria, and the host itself can activate this AIM2 inflammasome. It also can function in case of the autoimmune response against self-DNA in systemic lupus erythematosus. Now, let's move to other members of the NLR family. We can consider them as generally considered to perform the cytoplasmic surveillance for the bounds or dams. The NOD1 and NOD2 can recognize breakdown products of the bacterial cell wall, and their signalosomes can ultimately culminate in the activation of necrosis factor kappa, and this drives the pro-inflammatory gene expression. Also, the mutations of NOT2 are associated with human inflammatory response or diseases like Crohn's disease. NOT3 and NOT4 still awaits a little clarification. For the NNOD or NOT5 or NLRX1, this one is a matter of debate till now, and recent reports position it as or within the mitochondrial matrix. But we can also say that many of them are poorly characterized till now. But the end of this is formation of caspase 1 and finally lead to inflammatory response and cell death. And this is what is really needed at the end of the inflammatory process. Get rid of the microbial motives. And at the end of this, thank you so much for listening, my dear professors.